Alrighty, so this is just a video talking about the Agar Cube Lab. It's about cell sizes. And so in class we took a little slab of this, is called Agar. And so we cut it, it's sort of like really, really thick, like Jello. So we cut it into cubes. And in the instructions I told you to cut it into a 1 by 1 by 1 centimeter cube, a 2 by 2 centimeter cube, and a 2 by 2 by 2 centimeter cube. So I have them ready here. And then we're going to put them into methylene blue. Then we're going to let them sit for about 15 minutes. So I'll pause the video and then we'll come back. And then I'm putting on gloves because methylene blue it stains stuff terribly. And so we'll put that, and so then I won't get it on my hands. So we're going to put these two in the beaker, the 2 by 2 by 2 and the 1 by 1 by 1. Just going to let them sit in there, and then just for ease and sort of making sure that I don't use too much methylene blue, we're going to put this into here. So, and I put my glove on backwards, so it looks a little funny. Just going to fill it up till it's covered. Okay. And now we wait some 15 minutes. And then I will be back. Alrighty, so we have been sitting for a little bit. So I'm going to get this out of here. It's super messy, so I'm going to put that down there. And then we'll be right back. I'm going to pour this down the sink. So hopefully we'll be able to see this on the camera. Hopefully, if not, I'll just describe you what we see. So I'm just going to cut this in a little cross section. Oh yay! And then I'm going to cut this one in a cross section. I'm going to cut this one in a cross section. So they bleed pretty quickly once I open them up. So hopefully it'll sort of maintain. So if you can see, sort of in the middle of this one, there's a white. It's still white in the middle. And then this one, it has not much white. And then this one's sort of bled through, but hopefully you can still see it. It's on the edges, but it's not in the middle. And so that's what I wanted you to see with those. And so now we have to take measurements on the border of it. And so using a ruler and trying your best to see sort of how far they are, I want you to see how far the border is. And so if we take it, we'll go over here. You might not be able to tell, but I'll just tell you what it says. So it is about 0.2. The border is 0.2 around those. And if I were to measure around this one or around this one or around the teeny tiny one, all that border is going to be about 0.2 centimeters because the agar is the same in all three shapes, and the methylene blue is the same for all three. It affected them in the same way. And so then that border, the rate of diffusion is constant. And so it's 0.2. So remember that number, and then we're going to do a little bit of math. I'm going to pause it again. Alrighty, so I'll try to do this so that you can see it. If not, I'll talk through it and hopefully it'll make sense. So our border was 0.2. And so down here, I'm going to draw the sketch right here. So this was 1 centimeter cube by 1 centimeter cube by 1 centimeter cube. So if I draw a little sketch, and then this one would be 2 by 2 by 2. It's a little bit bigger. Just a cube. And then this one was smaller, 1 by 1 by 8. And so if we just draw those, my 7th grade art class is paying off. And so this is my 1 by 1, my 2 by 2, and my 1 by 1 by 8. And so then when we have this paper says pink, and it talks about acids and bases. The one that I have linked onto Schoology has the correct words on it, so these columns might be slightly different. And then hopefully my drawings will make sense. So I'm just going to draw where sort of it went in. Now ours was opposite of this because the inside was lighter and the outside was the blue, but I think you get the point. And so if we need to do the calculations, this is what we're going to sort of think about. And so this edge was 1, the whole thing was 1, and then this little bit was 0.2 and this little bit was 0.2. And so I'll come back to that in a sec, but we're going to do a little bit of calculation. So total cube volume is length times width times height. So this is 1 times 1 times 1, so it's 1 centimeter cubed, 2 times 2 times 2, so this is 8 centimeters cubed, and 1 times 1 times 8 is 8 centimeters cubed. So that's pretty easy. Surface area is if you take a cube, you take the area of the side times 6, and so the surface, or this 1 by 1 would be the sort of one side would be 1 by 1, so that's 1 times 6, so 1 side, 2 sides, 3 sides, 4 sides, 5 sides, 6 sides, and so then that would be 6 centimeters squared. And then a 2 by 2 would be 2 by 2 is 4, and then there's 6 of those because it's a cube, and so that would be 24 centimeters squared. And then if this one is a little bit different, so this would be a 1 by 1 plus a 1 by 1, so that's 2, and then I have a 1 by 8 times 4. So I have 1 plus 1 plus 8, 16, 32. 32. So this is 34 centimeters squared. And so we can do, without any of the math that we're going to not worry about right this second, we can do a surface area to volume ratio. And so I have surface area here to volume ratio over here. And so this one would be 6 to 1 because that's pretty easy. And then the next one is 24 to 8. And so this you need to minimize. And so 8 goes into 24 3 times. So this is a 3 to 1 ratio. And then I'm going to get my calculator because I forget what the exact number is. And then if I take 8 into 34, it is 4.25. So it's not perfect, but I want you to sort of write that number there because that will make sense when we talk about the ratio in a second. And so if we talk about surface area to volume ratio, the bigger the ratio, the better the cell. And so this smaller one is better than this bigger one. It is 6 to 1 to 3 to 1. So this cell should diffuse better. It should, as far as like getting rid of wastes and taking in nutrients, this one should be more efficient, but we're going to mathematically prove that. And so now we have the volume that was not pink. And so this would be the area that was blue. So sorry, I messed up with all the chemicals and stuff. The agar, I was about to throw it in the trash because it was driving me crazy. So now I have a cube, and the area that was not pink or was not blue is 1, the whole side, minus 0.2 minus 0.2. So this is 0.6 is the edge of this. It's a cube, and so if we take 0.6 times 0.6 times 0.6, then we have 0.21 is what this, 216. And so if I have the area is 0.216 that is not diffused, then I have to figure out the amount that was diffused through. So I take my total volume, so I take that number and I subtract it from 1. And so I get 0.784 is how I get that one. And then I have, this would have been 2 across here, minus 0.2, minus 0.2, so 1.6 would have been my my edge over here. So then if I take 1.6 times 1.6 times 1.6, that gives me 4.096, and I'm going to subtract it from this. And so I'll do that on my calculator. And so then I get, that's not right, 8 minus 4.096. So I get 3.904. And then the last one is, this would be 0.6 times 0.6 times 7.6. So hopefully that makes sense. You're just subtracting your little bits off of there. Times 0.6 times 0.6. So then this gives me 2.096. 736, and I subtract that from 8, because that's here is what I'm doing. And then that gives me 5.264. And so then it says the percent diffusion. And so if I have the numbers, and I think I did my math wrong. But if I have 0.7, no, I'm right. Okay. Got nervous there for a second. 0.78 divided by 1, then that is 78% diffusion. And then if I take 3.904 divided by 8, it gives me about, and then times 100, I get about 49% diffusion. And then if I take 5.264 divided by 8, I get about 66% diffusion. And so what this means is the higher the surface area to volume ratio, the better the cell. The more efficient the cell, the more diffusion it will be able to do more efficiently. And so then these numbers are proving that our smallest and our cube is the best. And then if we have the same volume, but we have different surface area to volume ratios, the one with the higher surface area to volume ratio will be better. And so an example of this is the cells in our stomach are long and skinny because their job is absorption. And so they increase their surface area, and then we get a better surface area to volume ratio because that's the function of those cells. And then, but we talked a little bit about cells are small. And then on the back, there's some questions. On the new one, this during is not in there because I took it out because the acids and bases, when I tried to set it up like that, it didn't work. But just try and answer those questions, and then hopefully you'll be able to answer them with the math that we did on the front. So hopefully that makes sense. I'll hold this up, and if you want to pause it and maybe copy down, hopefully all my math makes sense. But it's just mathematically and sort of observationally proving that surface area to volume, the higher the surface area to volume ratio, the better when it comes to cell size.